And the last short talk before a new time for questions and answers is focused on this time a startup. It's Mr. Stephane Boissel from Sparing Vision. You're going to talk about pioneering genomics to save sight.
Good morning. And this is the very embarrassing moment where I realize that my presentation is probably a misfit because I'm not going to speak too much about science, but more about business. How much time do I have? I was told 4 minutes. Is it more? Is it less?
Between 7 to 10.
Oh, plenty of time. Anyway, so Sparing Vision was started in 2016 as a spin-off from the Institut La Vision in Paris, and more precisely from the very fertile brain of Professor José Sahel, who is the scientific founder of the company. And the company is about doing or using genomic medicine tools in the space of ophthalmology. And we are more precisely focusing on in vivo. So we are not working on cell therapy, and genomic medicine and ophthalmology is the only thing we do. The company was started on the business principle of what we call a build-up. So we started with one asset, and through acquisition and licensing, we've built a portfolio of 6 products currently in the portfolio, gene therapy product and CRISPR-based product. The lead product, the one we started the company with, is now in clinical development, and all the pathologies that we address have very significant unmet need. We are working mostly or primarily today in what we call retinitis pigmentosa, which is the most prevalent form of inherited retinal disease, but we are going to expand in more prevalent disease, even more prevalent disease such as dry AMD or GA, for which there is, again, a very significant unmet need. In 2021, we entered into a strategic partnership with Intelia. I think in this room, everybody should know Intelia, the world leader in vivo genome editing, and probably the company that will be the first to have an in vivo CRISPR-based product approved, hopefully. And that partnership really put us on the map of the space. We have, as I said, one first product that started clinical development a year ago. And we have a second one that should start clinical development later this year or early next year. And so we expect 2 clinical readouts within the next 2 years on 2 very unique first-in-class gene therapy product. So I said I was not going to speak about science, but yet I will say a few words because what we are doing in gene therapy is unique. Most gene therapy approaches in the eye or elsewhere, by the way, are gene-specific. In our case, we are gene agnostic. So we are developing product, and we have 3 gene therapy products in the portfolio that you can see on the slides. They are expected to work independently of what has caused the mutation to start with. And that has many advantages. The first one is that theoretically, you don't need to genotype the patient to make him eligible for treatment. I should not say that because my scientists will yell at me. So yes, of course, we are going to genotype all patients, but theoretically, we don't need to do that. And very importantly, which is less of a concern for the audience today, but it's a significant concern for the investors in general, is that when you go one gene after the other, especially in the space of ophthalmology, the perspective of a return on investment is very small because the indications are very tiny. And the best example is Luxturna, which is a beautiful product that works very well. But at the end of the day, the market for Luxturna is very small. And so it will be very difficult today to reproduce what has been done with Luxturna because we are in a context where money is more difficult to find. So with our kind of approach, we will have more or less the same pathway to develop the product, more or less the same cost of development, but we expect much more significant commercial return should the product or the biology work. And in the case of the first product, and that's the only scientific spin I will give to the pitch, is that we have identified, I did not, but José Sahel has identified 20 years ago a neurotrophic factor. So he identified a gene. He split that gene into 2 isoforms, and those 2 isoforms are in an AAV gene therapy product, and that is the product that we are going to inject subretinally in patients to make sure or to try to preserve what is left of the vision at the time of intervention. So it's not typical gene therapy in a sense that first it's not gene specific, but also it's not aiming at replacing anything. It's aiming at protecting the cones through an action on their metabolism. So it's like a neurotrophic factor. It is a neurotrophic factor. So that's the first product currently in clinical development. And the second product is a product we bought from another spin-off from the Institut La Vision. It's also gene agnostic, but here we are not going to aim at preserving what is left of the vision at the time of intervention. So it's a different biology, a different transgene. We are going to aim at restoring function in cones that have lost their ability to function as part of the evolution of the disease. And the rest of the portfolio is less advanced. The first product that we developed with Intelia, the first CRISPR-based product of our portfolio, is probably, let's say, 3 years away from clinical development. And we are not disclosing the target, but it's, again, in retinitis pigmentosa. Here we are gene-specific, but we are mutation agnostic. And the idea would be in that target to use CRISPR to do single allele repression of a specific gene target in RP. That's very nice, very elegant. But if you believe gene therapy is complex, CRISPR is, let's say, leveling up the complexity to something totally unbelievable. And to make it work, we need a team of people. I'm not going to go through the resume of all these guys, but I'm only going to name 2 of them. Dan Chung, our Chief Medical Officer, joined us from Spark 2 years ago. And at Spark, he was instrumental in the development and launch of Luxturna, and he's now the Chief Medical Officer of the company. And every day that he's passing, I'm blessing Dan for him, being with us on this journey. And the second one is Mehdi Gasmi. Mehdi was educated at Geneton, and then he spent more than 20 years in the US. He was previously at Adverum, and Adverum, which is, by the way, going to announce today phase 2 data on their lead product, which is a gene therapy product developed in wet AMD, could be one of the first, if not the first, company to launch a product in that space. So we really have put together a team of experts in our space, independently of where they are coming from. We have an office. I mean, the company is headquartered in Paris. We have an office in Philly. We have 3 persons in California, 2 persons in Boston. And so the idea of that build-up strategy that was not only to find the assets wherever they are, as long as they fit the purpose, but also to put together the best possible team to succeed. And my last slide is about money. Of course, to do what we do, we need money. It's very costly. I like to say that from the patent to IND readiness, developing a gene therapy product or a CRISPR-based product, for that matter, takes $25 million, more or less. And then to get to proof of concept in man, you probably have to add something like 50. So 75 million per candidate. We have 6 candidates in the portfolio. Math is very simple. We need a lot of money. And we were very fortunate to raise a lot of money, not as much as what we need or we want to, but for a startup company in France, that was not too bad. We've raised 135 million euros since the beginning with the support from the shareholders listed in this slide, including BPI. And with that, I'm going to conclude my presentation. I think I went over the budget you gave me, but I'm sorry for that. Thank you very much.
Okay, thank you very much, Stephane.